Hello students and viewers. Today I am going to explain structure of DNA. So we know that DNA, here you can see, let me take the pen. This is the central axis. Here you can see this is the name of it. This is the black colored central line. This is central axis, right? On this central axis, this DNA is moving, or DNA is there, it is present in a helical like structure, right? So the diameter of the DNA is 20 angstrom. See here, this is the first characteristic of this DNA. So diameter, how much it is? 20 angstrom is the diameter of our DNA, right? Now here you can see whatever this double structure you are observing. See, this is the double structure. Let me change the color. See, see here, see at my pointer. So here these are the main backbone of DNA, right? So this backbone is made up of sugar and phosphate, right? Backbone is made up of sugar and phosphate, and these two backbone, they are running anti-parallel, right? Running anti-parallel. One is moving in this direction, right, which will going to start from this, and another will start from this direction, likewise, right? So likewise this will run anti-parallel to each other, right? So here you can see this is the backbone, which is made up of sugar and phosphate, right? So this, when these two, they are going to turn further, so during that turn they will going to form these grooves. Okay, they will form these grooves. One is the major groove. See, here is the name major groove, and another one is minor groove, right? So minor groove, as it's form very little groove, okay, little depth like structure. So that is the minor groove, which is of 12 angstrom, right? So minor groove of 12 angstrom, and major groove, here you can see here it is a 22 angstrom. So major groove, here it is mentioned in a blue color, you can see here. So this is actually the major groove, right? This entirely, this is the major groove, whereas this, you can see this is the minor groove. So likewise here major groove, here again minor groove. So likewise these major grooves and minor grooves are present. Minor groove of 12 angstrom and major groove of 22 angstrom, right? So here you can see the complete turn. It is shown in the green color. See this arrow, right? Let me take this again, highlighter, laser pointer. Here you can see this is the complete turn in which the DNA has its major groove, this, as well as DNA has minor groove. And complete one turn, it is of 34 angstrom, right? Or we can say in a nanometer it is 3.4 nanometer, right? It is in length of one ton of DNA, right? So complete one ton has major groove, minor groove of 12 angstrom, major groove of 22 angstrom, and the complete length of this complete turn of DNA is 34 angstrom, right? And if you see this central, this ladder like structure, right? So these structures, these are actually the nitrogenous bases. So what are the nitrogen bases? I will explain in my further slides, right? So in this diagram, I want to clear what is the actually major groove, what is actually minor groove, what is the distance of it, and what is the distance of complete one turn of DNA. Now, so here you can see here now, this is the in a blue color, this is the sugar phosphate backbone. See here the name, right? So this is the sugar phosphate backbone. As I explained, these are anti-parallel to each other, and whatever now in a centrally, whatever you are observing, see this. So these are actually base pair. So this is the nitrogenous basis. Okay, all these are what? Guanine. G stands for what? Guanine. C stands for cytosine. T stands for thymine, and A stands for adenine. So likewise, these are the guanine, cytosine, thymine, adenine. These are the four nitrogenous bases which are present in a DNA, right? Only in the RNA, this T is replaced by uracil, right? So uracil is there in a RNA, right? Rest of these nitrogenous bases are similar. Getting? So all these nitrogenous bases, they are present in a DNA. Okay, in my further next slide, I will explain you which kind of, there are again two main types of these nitrogenous bases. Now, see here, again see how the DNA looks like and how RNA looks like. So RNA is nothing but what? Ribonucleic acid, whereas DNA is deoxyribonucleic acid. DNA has two these backbones. Okay, two these backbones of sugar phosphate, whereas RNA has only one. Or in a simple language, or in a simple this sentence, I can say RNA has only one strand, whereas DNA has double strand. Okay, any this word you can use. Okay, two backbones or one backbone, or two these strands or one strand, right? Only do remember what this backbone is made up of, right? So backbone or a strand is made up of what? It is made up of, let me take a pen. It is made up of, as I said earlier, it is made up of phosphate and sugar, right? It is made up of phosphate and sugar, and whatever this structure, they are made up of these nitrogenous bases, right? So here I have shown you, C stands for cytosine. Okay, here it is C, this is C. Then G stands for guanine. Again see here, it is a guanine structure, right? Then A stands for adenine. Here you can see this is the adenine. Then T stands for thymine, right? So again see here thymine. So always do remember there is again a particular manner in which they are attached to another these nitrogenous bases. So A is always going to pair with T, and C is always going to pair with G, right? So here only see the difference between this DNA and RNA. See, cytosine is similar, here is also similar, right? Guanine also similar, here is also similar. है ना? Next adenine, this is also similar. Here is also similar. This is similar, cytosine also similar. Only difference is this. Thymine is present in a DNA, whereas uracil is present in a RNA. No thymine is present in RNA, right? So this is the main difference you have to remember in case of RNA. Please do remember, uracil is present. Uracil is one of the nitrogen base. It is present only in RNA. Getting? So that is the difference between this DNA and RNA, right? Next come DNA versus again RNA. Now here we can see Let me change the color. Okay, so here see again one strand, as I explained you earlier. These are again two strands in a DNA. Here one strand in a RNA. Now here see what is the main difference here. A is joined with U, where in case of RNA, A is joined with uracil with double bond. In case of RNA, see always rule is what this. Okay, let me uh, tell you first this A and G. A is nothing but what? Adenine. G is nothing but guanine. These are purine bases. These are what? These are purine bases. Whereas these cytosine and thymine, these are pyrimidine bases. These are what? These are pyrimidine bases, right? So always rule is what? Rule is, I will write here, purine is going to join with pyrimidine bases. Purine is going to join with pyrimidine bases, right? So this is the rule, right? So here see adenine from the purine is going to join with thymine of pyrimidine, right, with two bond. How many bonds are here? Two hydrogen bonds, right? Adenine going to join with T with two hydrogen bond, whereas guanine of the purine, listen me carefully, guanine of purine going to join with cytosine of pyrimidine, right? With how many? With three hydrogen bonds. These are what? Bonds are made up of hydrogen, right? So two hydrogen bond between whom? Between adenine and this thymine, and three hydrogen bond between guanine and cytosine. Please do remember, this is very basic knowledge you have learned earlier in 12th, and again you are repeating here in FYBSc, right? So guanine with this cytosine with three hydrogen bond, and adenine with thymine, right? So only difference what we are observing in case of this RNA, adenine is going to pair with whom? Uracil, right? Adenine is going to pair with uracil because uracil is present in RNA. Okay, thymine is not present in RNA. It's absent in RNA, right? Whereas uracil is absent in DNA, vice versa. Please do remember. And rest of this bond, that is two hydrogen bond, three hydrogen bond, similar, right? In case of this RNA again. So this is the main purine and pyrimidines which are present inside these two strands. Come to the next. Now this is the main part. This is the main part, that is nucleoside and nucleotide, right? So what is nucleoside? So when pentose sugar plus nitrogen base, right? Suppose I write here, this is the sugar. It's symbolically it's written as S. Suppose it's going to join with our A, adenine, which is join with double hydrogen bond with T. Okay, suppose these two, let me change the color. These two are what? These two are nitrogen base, right? And this is our pentose sugar. So when this only these two structure are present, then we called it as what? Nucleoside, right? When see here, same is here. Pentose sugar is here. Also nitrogen base is also here. Only addition is what? This is the phosphate group. So if pentose sugar along with the nitrogen base along with this phosphate group, then we have to call this as a nucleotide. Please do remember, nucleotide is made up of sugar, nitrogen base and phosphate group, whereas nucleoside where phosphate group is absent. Only pentose sugar and nitrogen base is present, right? So this is the nucleoside and nucleotide, right? Suppose these are the two strands. Getting? So here what we have learned right now, this strand is made up of what? Phosphate and sugar, right? And here internally these are the bases they are present. A is going to pair with T, then C is going to pair with G, right? So these are what? Nitrogenous bases. These are nitrogenous bases, right? So when nitrogenous bases going to combine with this phosphate as well as this sugar, then it is all these three, they are said as nucleotides. If this phosphate group is absent, only this nitrogen base and sugar is present, then it is called as what? Nucleoside. Right? Next. Then again see here nucleoside, nucleotide in a diagrammatic way. I have explained this. So here again see this is the main base. This is your sugar, then you have to call it as nucleoside. When this base, the sugar, and then this is the phosphate, all these three are present, then we have to call it as nucleotide, right? Nucleotide consists of three things, whereas nucleoside consists of two things only. Two things, base and sugar, right? Here I have mentioned base and sugar, we called it as nucleoside, whereas nucleotide, base, sugar and phosphate group, all these three together we called it as nucleotide. Then see, so again we have to learn one by one. First, sugar. So here we know that there are two types of sugars. They are present, deoxyribose sugar whereas ribose sugar, right? Deoxyribose sugar and here ribose sugar. So what's the difference between these two? So see, rest all these structure are similar except this. Here you can see, here it is in a second carbon atom OH is attached, whereas in a second carbon H, only H hydrogen is attached. Okay, so this is the main difference between these ribose and deoxyribose. Okay, so sugar is again see 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. So likewise 5 carbon atom containing this sugar is pentose. Okay, both these are what? These are pentose sugar, right? 5 carbon containing this sugar are. Here is also you can see first prime, second prime, third prime, 4 prime and this is the fifth prime. So all these prime, all these carbon they are numbered as a prime, right? So 5 carbon containing sugar, hence we called it as what? Pentose sugar. Only the difference between the second carbon containing of hydrogen or OH group, right? So on that this sugar is differentiated as deoxyribose and ribose, right? So in a second carbon the OH group is attached in case of ribose, in case of deoxyribose H is attached to the second carbon number, right? So all it's clear, rest of the structure is similar, you can see this structure. Now see nitrogenous bases. So here we know that nitrogenous bases there are total 5. How many are there? Total 5. So here guanine and adenine, these are purine bases. Student, please do remember, guanine and adenine, these are purine bases, whereas cytosine, thymine and uracil, these are pyrimidine bases. Okay, these are pyrimidine bases, right? Cytosine, thymine, these two they are present in whom? They are present in DNA, right? Whereas cytosine and uracil, this is present in RNA, right? So these are three are pyrimidine bases, these two are purine bases. And you can see the internal chemical structure. Now see again in I have explained this with another slide. This see guanine, adenine. What I said, these are purine bases because they are double ring structure. You can see ring number 1 and ring number 2. So here is also 1 and 2. So here double ring structure of these purines. So purines are guanine and adenine. These are present both in DNA as well as in RNA. Okay, both these adenine, guanine, they are present in DNA as well as in RNA, right? So these are double ring structures, right? Now see the next, that these are the cytosine, thymine and uracil. Here we are not observing any structure, right? So only here it is what single ring structure, right? So here they are single ring. There is no any another ring adjacent or any attached to it, right? So these are the single ring structure pyrimidines are. Now here you can see cytosine is present both in DNA RNA, whereas thymine, now see, please do remember thymine present only in DNA, whereas uracil, this is present only in RNA, right? So this is the basic difference between these uh, pyrimidine bases present in DNA and RNA, right? Now, next is phosphate group. Now, from where this phosphate group arises? Always what we are saying, this backbone is made up of what? Phosphate and sugar, right? We are always saying this is this part, this particularly, है ना? These are the bones which are made up of phosphate sugar, right? So this phosphate group is coming from phosphoric acid. Okay, so this phosphate group coming from whom? Phosphoric acid. So see, the molecular formula of phosphoric acid is H3PO4. This is the molecular formula, right? So H3PO4. So this is the hydrogen number 1, 2 and 3, H3. Then O4, 1. Again see, this is the one. Let me change the color. This is oxygen 1, this is 2, then this is 3 and this is 4th, right? So likewise H3PO4 is there and it contains 3 monovalent hydroxyl group. See, here it is only single strand is present, monovalent, right? These are monovalent hydroxyl group and a divalent oxygen atom. Here you can see double bond structure. Okay, so this is a divalent. Okay, divalent oxygen. This is divalent. Double bond is present between this phosphate and phosphorus atom and this oxygen, right? So all linked to pentavalent phosphorus atom, right? So likewise this phosphoric acid, so when this hydrogen atom is removed from this phosphoric acid, then structure comes, that is our phosphate group, right? So as what I said, phosphate group is coming from whom? Phosphoric acid. And when we call phosphoric acid as a phosphate group, when this hydrogen atom will removed from this phosphoric acid, then phosphate group is formed, right? So here you can see this hydrogen atom is removed and hence the negative charge is coming on this oxygen, right? So likewise this phosphate group is coming from phosphoric acid. Now see, here see, again see, here it is, this is the phosphate group, right? This is the our sugar and this is nitrogen basis, right? So particularly adenine is shown in this figure. So this is our nucleotide. This is what? This is nucleotide, right? So nucleotides, they are present in a DNA, right? And nucleoside, when this phosphate group is not present, only this sugar and nitrogenous base are there, then we have to call it as nucleoside. So likewise, we have seen this structure in detail. So thank you very much for this watching this lecture and listening this lecture. If you have any doubt in the structure or any doubt in the nitrogenous bases, or if you don't understand what are exactly purine pyrimidines, then please do call me, or you can have a mail to me. My mail ID is there in a WhatsApp group, right? As well as on a Google Classroom. You can ask me directly. Thank you very much for patience listening.